Hello, my name is Robin Haw and I work at the Ontario Institute for Cancer Research, and today I'll be talking about the Reactome project. Reactome is an open source, open access pathway knowledge base. We provide expert authored, manually curated and peer reviewed pathway modules traceable back to the primary literature, and we've developed user-friendly, biologist intuitive software tools for data visualization and integration and analysis. The central component of our pathway curation is our data model, which is based upon the reaction. The flexibility of this model allows us to capture many different reaction types in Reactome, and we can even capture drug-target interactions. Typically, the drugs we curate bind their target, and the resultant complex regulates the target reaction. As a pilot study, we focused on the annotation of cardiovascular drugs to work out the curation process and to make sure this approach fits with our data model. So reactions are then grouped into ordered causal chains to form pathways, and these pathways in turn can be grouped to represent human recognizable biological events. Now to date, we've also detailed the information on altered pathways corresponding to human diseases, and we've curated over 2,000 variant proteins and their involvement in disease pathways. The pathway browser is the main visualization tool to browse biological pathways, and we provide different types of pathway visualizations. In this pathway browser widget, the pathway hierarchy is in the left panel, the diagram view is in the right, and a details panel below the diagram provides information as the user interacts with the pathway diagram. The pathway browser provides a framework for data analysis and visualization of external datasets, both user-supplied experimental data and data provided by other online resources. The pathway overview shows a visual, interactive summary of the results of the pathway analysis. The list of significant pathways is listed below the diagram, and as you drill down into specific pathways, you can see the specific textbook illustrations or pathway diagrams with the experimental data overlays. Now, new to Reactome is this high-level pathway overview visualization based on the Voronoi tessellations. To support multi-omics data analysis, we have a new Reactome GSA service that uses our existing web interface and a novel R bioconductor package. For multi-omics gene set analysis, Reactome GSA supports the direct import of data from the Expression Atlas as well as user-supplied data, and offers three different analytical methods. The first being camera through the Lima package, the second being PADOG, and the third is GSVA, which provides support for single sample gene set enrichment analysis. For single cell RNA-seq pathway analysis, the Reactome GSAR package will support the direct importation of data from the single cell expression atlas, as well as user supplied data. The Reactome project also provides a suite of tools called the Reactome F5Vis App for our users to perform single and multi-omics pathway and network-based data analysis via the Cytoscape platform. Following a recent update, the functional interaction now consists of 13,000 proteins and 436,000 interactions. The Reactome F5Vis App now provides support to perform gene set enrichment analysis for Reactome pathways using a gene score file. Users can also overlay drugs onto Reactome pathways and networks, including FDA-approved cancer drugs. And users can also perform different types of simulations to predict the pathway activities subject to a drug or genetic perturbation. Now, as some of you know, Python is becoming more and more popular in the data science in general and bioinformatics in particular, and to support our own and other data analyses and visualization projects, we've developed a Python package by wrapping our APIs. These APIs are the analysis service, the content service, and the services associated with the Reactome F5Vis App. We've also developed the documentation and deployed these packages into several Python repositories, and have also provided a Docker file. So now I'd like to talk to you about the project that we're working on with the IDG, or Illuminating the Druggable Genome project. The goal of the IDG project is to catalyze research to improve the understanding of properties and functions of understudied proteins within commonly drug-targeted protein families. Reactome is developing a portal by integrating resources collected in the IDG project and other places to provide a pathway-centric view for understudied human proteins. Before I show you our progress to date, I'd like to introduce you to the IDG protein classification scheme called the target development levels, and there's 4 categories. T-clin represent the protein targets that have at least one approved drug. T-chem represents the targets that have at least one chemical compound. T-bio represents the targets that don't have any known drug or small molecule activities but have some publications and experimental data. And finally T-dark, and these are the targets about which virtually nothing is known. This is a screenshot showing the diagram for G-alpha signaling pathway looks like after overlaying the target development levels. We can see the 4 IDG development levels associated with the Reactome entities. Taking a moment to explain the color overlay, dark blue colored entities represent the T-clin level, light blue for T-chem, orange for T-bio, and red for T-dark. Now we can zoom into the diagram to view the olfactory receptors entity, and we can see that these contain a number of T-dark proteins. Now in the past, we would have had to have performed some pathway enrichment analysis to overlay this kind of experimental data onto the pathway diagrams. The portal also supports the overlay of protein and mRNA expression data collected in the IDG project. This screenshot shows 2 human proteome map protein expression data sets for adult heart and adult liver in the pathway called ion channel transport. The focus is on one reaction, and on average the proteins involved in this reaction has higher expression in the heart than in the liver. Users can toggle to the simplified FI view, which shows clearly that RYR2 and 3 and several other proteins have higher protein expression in the heart than in the liver. And we've also implemented features to overlay pairwise relationships together with target development level information. And in this table, we can see the drugs that are known to interact with members of the RYR tetramer complex. So in summary, Reactome is a highly reliable curated database of biological pathways. Through our website, we provide tools and data sets for visualizing pathway data and interpreting your own experimental data. All our data and software is open source, open access. The Reactome GSA platform greatly reduces the technical barriers for multi-omics, cross-species comparative pathway analyses. The Reactome F5Vis App provides a very powerful way to analyze and visualize single and multi-omics data sets. And the Reactome IDG portal will hopefully provide a pathway-centric view for understudied human proteins. And finally, just take a moment to acknowledge the valuable contributions of the Reactome Consortium members and the support for our funding agencies. Thank you.